Today, let's talk about Chapter 6, The Developing Brain. The neurons develop through delicate and carefully processes that take place while an embryo grows. Signaling molecules turn on certain genes and turn off others, initiating the formation of immature nerve cells. During the next stage, cell division and also called proliferation, the pool of early-stage brain cells increases by billions. Finally, during migration, these newly formed neurons travel to their final destinations. The nerve system formed by this process is active throughout life, making new connections and fine-tuning the way messages are sent and received. In this chapter, you will learn about the amazing early development of your ever-changing nerve system and the journey of nerve cells, including several processes. First, formation and induction. During the very early stages of embryonic development, three layers emerge: the ectoderm, the, the outermost layer, and mesoderm, middle layer, and endoderm, innermost layer. Although the cells in each layer contain identical DNA instructions for development, these layers ultimately give rise to the rich variety of tissue types that make up the human body. The explanation of this diversity lies in signals produced by surrounding tissues. Those signals turn certain genes and others off, thus inducing the development of specific cell types. Signals from the mesoderm trigger some ectoderm cells to become nerve tissue, a process called neural induction. Subsequent signaling instructions refine the nerve tissue into the basic categories of neurons or glia, and then into subclasses of each cell type. The fate of a developing cell is largely determined by its proximity to various sources of signaling molecules. The concentration of each type of signaling molecule decreases further from its source, creating gradients throughout the brain. For example, a particular signaling molecule called sonic Hitchcock is secreted from mesodermal tissue lying beneath the developing spinal cord. As a result of exposure to this signal, adjacent nerve cells are converted into a specialized class of glia. Cells that are further away are exposed to lower concentrations of tonic hedgehog. So they become motor neurons that control the movement of muscles. An even lower concentration promotes the formation of interneurons, which don't relay messages to molecules but to other neurons. Interestingly, the, me- the, mecha- the mechanism of this molecular signaling is very similar in species as diverse as flies and humans. And the next step is called proliferation. In the brain, neurons arise from a fairly small pool of neural stem and progenitor cells, special cells that can divide and become a variety of mature cell types. Before achieving their mature cell fate, this pool of cells undergoes a series of divisions, increasing the number of cells that will ultimately form the brain. Early divisions are symmetric. The split results in two identical daughter cells, both able to keep dividing. But as these divisions progress, the cells begin to divide asymmetrically, giving rise to only one daughter cell that keeps proliferating and a second that progresses towards its ultimate cell fate as a neural or glial cell. This proliferative process permits rapid growth during early development of the brain, with billions of cells being produced in a matter of weeks. After this series of divisions is complete, only a few neuron stem and progenitor cell remain within the brain, and neuron genesis in adulthood is limited to a few regions of the brain, such as those involved with memory. Scientists have proposed that protein defect causing a premature switch from symmetric to asymmetric divisions may be a cause of microcephaly. This disorder, characterized by a severe reduction in the brain size, is associated with serious neurological disabilities and sometimes death in infancy. Similarly, excessive proliferation of brain cells can lead to a disorder called megalencephaly, a brain that is abnormally large and heavy, which is also associated with a variety of neurodevelopmental complications. And the next step is called migration. After neuron induction and proliferation occur, new neurons journey from the inner surface of the embryonic brain, where they formed, to their long-term locations in the brain. And this process is called migration, and it begins 3 to 4 weeks after a human baby is conceived. At this time, the, the ectoderm starts to thicken and build up along the midline of the embryo. As the cells continue to divide, a flat neural plate grows, followed by the formation of parallel ridges, somewhat resembling the creases in a paper airplane, that race along either side of the midline. These ridges extend from the head end, where the future brain will form, along the length of embryo where the future spinal cord will develop. Within a few days, the ridges fold together each other and fuse into a hollow neuron tube. The head end of the tube thickens into 3 bulges that form the head brain, the midbrain, and the forebrain. Later in the process, at week 7 in humans, the first signs of the eyes and the brain's hemispheres appear. As new neurons are produced, they move from the neuron tube's ventricular zone, which lies along the inner surface of the tube, towards the border of the marginal zone, or outer surface. After neurons stop dividing, they form an intermediate zone where they gradually accumulate as the brain develops. A variety of guidance cue neurons to migrate to their final destinations. The most common guidance mechanism, accounting for about 90% of migration in humans, is the radial glia, which project radially from the intermediate zone to the cortex. Neurons use this glia as scaffolding, inching along glia projections until they reach their final destinations. This process of radial migration occurs in an inside-out manner. That is, the cells that arrive the earliest form the deepest layer of the cortex, whereas the late arriving neurons form the outermost layer. Through a different mechanism, other neurons migrate sideways, or, or tangentially, mo- or tangentially, moving parallel to brain surface and across the radial cortical columns. Migration is a finely tuned process that can be influenced by many factors. For example, exposure to alcohol, cocaine, and radiation can prevent proper migration, resulting in misplacement of cells, which can lead to the intellectual disability or epilepsy. Furthermore, mutation in the genes that regulate migration have been shown to cause rare genetic forms of intellectual disability and epilepsy in humans. And the next step is making connections. After neurons each reach their final locations, they begin making a connections that will determine how particular functions, such as vision or hearing, can occur. Induction, proliferation, and migration occur internally during fetal development, but the next phases of brain development depend increasingly on external experience. After birth, factors such as watching a mobile screen, listening to voice, and even proper nutrition influence the connections formed by neurons. And neurons become interconnected through their short branches called dendrites and long axons, two types of process that extend from a neuron's cell body. And then the synapse formation. Once axons reach their targets, a specialized connections called a synapse begin to form. At the synapse, only a tiny space separates the signaling portion of the axons from the receiving portion of the dendrite. Electrical signals that travel down the axon trigger the release of chemical messages called neuron transmitters, which diffuse across the space and are received by receptors on the target dendrite. Such chemical cues can either promote or hinder the generations of a new electrical signal in the receiving neuron. The combined effect of such cues from thousands of synapses ultimately determine how a receiving neuron responds. The human brain contains trillions of these synapses, which gives rise to the brain's outstanding capacity for information processing. And the next step is myelination. Insulation that covers rice preserves the strings of electrical signals that travel through them. The myelin sheath that covers axons serves a similar function. Myelination, the fatty wrapping of axons by extensions of glia, increase by as much as 100 times the speed at which signals can travel along axons. This increase is a function of how the sheath is wrapped, with somewhat regularly spaced gaps called nodes of reservoir interrupting the sheath. The alternating pattern of insulation and nodes allows electrical signals to move down an axon faster, jumping from one node to the next. This phenomenon called sati- saltatory conduction is responsible for more rapid transmission of electrical signals. Formation by myelin occurs throughout the lifespan. And the last step is called pairing back. After initial growth, the neural network is paired back, creating a more efficient system. In fact, only about half the neurons generated during development survive to function in an adult. Entire populations of neurons are removed through apoptosis, a process of programmed cell death initiates in the cells. Apoptosis is activated if a neuron fails to receive enough life-sustaining chemical signals called trophic factors, which are produced in limited quantities by target tissue. Each type of trophic factor supports the survival of a distinct group of neurons. For example, nerve growth factor is important for the survival of sensory neurons. It has recently become clear that apoptosis is maintained into adulthood but constantly held in check. Based on this, researchers have found that injuries and some neurodegenerative diseases kill neurons not by directly inflicting damage but by activating the cells when death programs. This discovery and its implication that death need not follow insult have led to new avenues of therapy. And just as too many brain cells develop early on, these cells initially form an excessive number of connections. In pyramids, for example, neuron projections from the two eyes to the brain initially overlap. Then, in some portions of the brain, they sort into separate territories devoted to one eye or the other. Furthermore, connections between neurons in a young primate cerebral cortex are more numerous and twice as concentrated as in an adult primate. A primate. The, the pruning of these excess connections is heavily dependent on the relative activity of each connection. Connections that are active and generating electrical currents survive, while those with relatively little activity are lost. Astrocytes and other glia also play an important role in this process. For example, astrocytes are known to aid the formation of eye-specific connections by engulfing and eliminating unnecessary synapses. Thus, at least to some extent, the circuits of the adult brain are formed by pulling away incorrect connections to leave only the correct ones. And that's all about this chapter.